I am here with another video in Dairy Pathshala that is very important topic in microbiology, gram staining. You can say in industrial point of view, microbial point of view, theoretical, practical, every point of view, this topic is very important. So you should know the each and every step and each and every mechanism of this topic. So you should see this video. Now I will explain step by step the procedure, mechanism, and some basics. Now see what is staining. Actually staining means we are using some dye. हम लोग क्या करते हैं डाई और कलर यूज करते हैं किसी भी चीज को अच्छे से देखने के लिए राइट कलर यूज करने से हम लोग उसको बहुत अच्छे से देख सकते हैं दैट्स व्हाई वी आर यूजिंग डाई टू सी एनी माइक्रो ऑर्गेनिज्म okay that process is known as staining then why we need staining why we are using this color to see those microorganisms actually हम लोग क्या करते हैं हम लोग माइक्रोस्कोप की यूज करते हैं राइट सो इधर क्या होता है इधर अपन का होता है लाइट सोर्स ये इधर से हमारा लाइट आता है अभी ये लाइट ऊपर की तरफ आ रहा है ये है इधर हमारा स्पेसिमन सो इधर है अपन का लेंस दैट लेंस विल हेल्प टू मैग्नीफाई दोज ऑब्जेक्ट्स और दोज इमेज सो दैट वी कैन सी थ्रू दिस आईपीस लेंस और थ्रू आवर आई बट इधर क्योंकि हम लोग बस लाइट सोर्स यूज कर रहे हैं बस लाइट सोर्स सो दैट विल मेक वन ट्रांसल्यूसेंट इमेज which is not properly visible. So if we stain that with some color, it will become more properly visible. So we are using staining process. Okay. That is why we are doing staining so that we can see this colorful picture of microorganism, right? Now, what are the types of staining? First, simple staining, differential staining, and spatial staining. I will explain this simple and spatial in my further video, but today I will discuss about differential staining, that is especially gram staining, because the use of gram staining is very frequent in dairy industry or especially in food industry. Okay. In my next video, I will explain the another type of differential staining, that is acid fast staining. Okay. So first differential staining. Why we are doing differential staining? Because to differentiate the gram positive from gram negative, we should do differential staining. हम लोग क्या करना चाहते हैं डिफरेंशिएट करना चाहते हैं ग्राम पॉजिटिव को ग्राम नेगेटिव से ग्राम पॉजिटिव ऑर्गेनिज्म को ग्राम नेगेटिव ऑर्गेनिज्म से that's why we are using this differential staining very simple so first one is gram staining so why this is known as gram staining because of Hans Christian Gram this person published this procedure in 1884 what is gram staining I already told because of this person that is Hans Christian Gram it is named as gram staining and why we need this gram staining to differentiate the gram positive organism from gram negative organism based on their cell wall characteristics so that they can absorb some color and we can see them with the help of some microscope or light microscope. Okay, that is the process of gram staining. Now see, gram positive bacteria at the end will show violet color and gram negative bacteria at the end will show pink or red color. Why I will show the mechanism in my further slide. Now see, first you should know before any procedure and mechanism, you should know the cell structure, cell wall structure especially. Actually, every bacteria is having outer layer that is cell wall, then cell membrane, then inside they are having cytoplasm, right? So this is the cell wall. So in gram positive organism, the cell wall is having peptidoglycan layer. So this peptidoglycan is very thick. See, but for gram negative, the peptidoglycan is very thin, very thin. For gram positive, the peptidoglycan is having teichoic acid. Teichoic acid is embedded in peptidoglycan layer. Okay, see, and this teichoic acid is negative in charge. Okay, negative in charge. Remember this. And why this is negative? Due to the presence of phosphate group in their structure, in teichoic acid structure. Okay, I will explain in more detail that cell structure in my further video for only for cell structure. Now you should only remember that it is having phosphate group. That's why the teichoic acid is negative in charge. Okay. For gram negative, no teichoic acid is present. No teichoic acid is present, but they are having this outer membrane, which is having some negative charge. Another outer membrane that is known as lipopolysaccharide, which is having lipopolysaccharide. Okay, lipopeptide layer. This is the lipopeptide layer, which is also having negative charge. Okay, due to the presence of that phosphate group only in this structure. Okay. So it is gram negative also having negative charge, gram positive also having negative charge, but here the negative charge due to the presence of teichoic acid, and here the negative charge due to the presence of lipopeptide layer. Here this is thick peptidoglycan layer, here thin peptidoglycan. Remember this. This differentiation is very important. You can see in exam point of view, in theoretical point of view. Okay, and to understand the mechanism too. Okay. Now see the procedure. This is the bacteria. We should first make the bacterial smear. Okay, this is our slide. Think the slide. First, we should make the bacterial smear, and we should add first crystal violet. Crystal violet, that is known as primary stain. What is primary stain? Okay, crystal violet. तो क्रिस्टल वाले जैसे ऐड किए हम लोग, क्या हुआ, दोनों ग्राम पॉजिटिव ग्राम नेगेटिव दोनों दोनों के पास पेप्टाइडोग्लाइकन है. अब वो अलग बात है कि वन इज हैविंग थिक एंड वन इज हैविंग थिन, वो अलग बात है, बट दोनों के पास है. दैट्स व्हाई both are taking violet color. Okay, so crystal violet, first step is crystal violet. We are adding crystal violet, then we'll wash the slide with water, and then we'll add grams iodine. See the time also. Okay, note the time in your copy. Time is also important for ICR exam point of view. They will ask. Okay, so the application of iodine, this is actually mordant, means it helps to fix the crystal violet color or crystal violet stain in gram positive bacteria. Okay, so now this is both are showing violet color. Then we will add decolorizer, that is ethyl alcohol ethanol. As we add decolorizer, the color of gram negative bacteria that is removed, but gram positive के पास अभी भी वो कलर रह जाता है. Okay, after adding decolorizer. So this is having no color, so we should add some color, that is some stain. So we are adding saffronin, that is pink in or red in color. Okay, that is also known as counter stain. So as we add the counter stain, the gram negative at last under light microscopy under 100x oil immersion, it will show pink color, and it will show gram positive will show violet, gram negative will show pink or red color. This is the procedure. So first you should add primary stain, that is the crystal violet. Then you should add mordant. Mordant is iodine. Then you should add decolorizer. Decolorizer is ethanol or ethyl alcohol. Then you should add counter stain. Counter stain is saffronin. PMDC. As I already told you, that gram positive is having thick peptidoglycan layer. See, and gram negative is having thin peptidoglycan. So first primary stain, that is crystal violet. As we add crystal violet is a basic type. So crystal violet is having positive charge. Okay, in their structure. And this peptidoglycan layer in gram positive organism is having teichoic acid. It is having negative charge. This negative and crystal violet positive, that will attract and that will give color to the peptidoglycan layer. Same in gram negative bacteria, it is having negative group in this lipopeptide layer due to the presence of phospholipid. So negative charge and crystal violet positive charge, that will attract and that will give color to the peptidoglycan layer. अब वो बात अलग है कि इसका थिक है इसका थिन है, तो इसका कलर थोड़ा कम है इसका कलर ज्यादा होगा ड्यू टू द एरिया. यू कैन से फर्स्ट दोनों वायलेट कलर दिखाते. देन वी आर एडिंग iodine, grams iodine, that is a mordant. Why we are adding grams iodine? To fix the color in gram positive or gram negative, anyone, to fix the color of crystal violet. Crystal violet की color को fix करने के लिए we are adding mordant, that is iodine. Then we are using decolorizer, that is ethanol. Why we are using the decolorizer? अभी जब भी हम डीकलराइजर ऐड कर देंगे, ये तो एकदम फिक्स हो चुका है ग्राम पॉजिटिव में, क्योंकि एरिया थिक एरिया, बट इधर वो फिक्स होने के बाद भी एरिया क्योंकि कम है ना, तो वो कलर निकल जाता है, तो वो डीकलराइज कर देते हैं. इथानॉल डीकलराइजेस दिस पेप्टाइडोग्लाइकन लेयर इन ग्राम नेगेटिव बैक्टीरिया. तो अभी ये कलर लेस हो चुका है, सो इसको कलर देने के लिए हम लोग क्या करते हैं, उसके बाद हम लोग काउंटर स्टेन ऐड करते हैं, दैट इज सैफ्रोनिन. सैफ्रोनिन जैसे ऐड करते हैं, ग्राम पॉजिटिव के पास ऑलरेडी कलर है, तो इसको इधर ही ऐड नहीं होता, इधर ऐड नहीं होता, तो ऐड कहाँ होते हैं, ग्राम नेगेटिव के पास ही ऐड हो जाता है. सो ग्राम नेगेटिव अभी कैसा कलर हो जाएगा, रेड या पिंक कलर. ओके, वी ग्राम नेगेटिव ऑर्गेनिज्म ऐसे दिखेगा, ग्राम पॉजिटिव दिखेगा वायलेट, ग्राम नेगेटिव दिखेगा रेड. सेम रीजन, सैफ्रोनिन is also a basic dye, so it is having positive charge, and gram negative cell wall में क्या है, phospholipid है, so negative, so positive negative it will attract each other and it will give color to the cell wall. So gram negative means red due to safranin, gram positive means violet due to crystal violet, that is primary stain. Okay, we should know this mechanism very properly. Okay, this is actually very much used in dairy industry. Okay, एंड दिस प्रिंसिपल मैकेनिज्म, और मैं जो वो टाइमिंग वगैरह भी बोली हूँ उधर 60 सेकंड वो भी नोट करके रखना, वो एग्जाम पॉइंट ऑफ व्यू से बहुत ज्यादा इंपॉर्टेंट होता है। विल एक्सप्लेन इन माय नेक्स्ट वीडियो दैट एसिड फर्स्ट स्टेनिंग। ओके।